In today's documentary, we are going to talk about the first human. And here is the first thing you need to know. There was no first human. Not in the way you think. Not a single individual. Not a single moment. Not a single birth on a single day in a single location where you could point to a creature and say, "There, that one." That is where we begin. That is not how evolution works. That is not how species work. And that is not what the fossil record shows us. What the science actually tells us is far stranger, far messier, and far more interesting than any clean origin story anyone has ever told you. Right now, inside your body, you carry the DNA of at least 2 other species of human being that are not Homo sapiens. Species that lived alongside your ancestors. Species that your ancestors had children with. And then, eventually, species that disappeared from the face of the earth while your ancestors did not. The bones of our earliest relatives have been found in Africa, crushed under millions of years of rock and sediment, preserved by nothing more than geological accident. Most of them were never preserved at all. Most of them rotted into the ground and left us nothing. The ones we have found represent a tiny fraction of the creatures that actually lived. And yet, from those fragments, from shattered skulls and broken teeth and partial femurs, scientists have reconstructed a story that stretches back 7 million years. 7 million years. To put that number in context, the entire recorded history of human civilization is approximately 5,000 years. The oldest writing we have found is approximately 5,000 years old. The oldest city we have found evidence for is approximately 10,000 years old. And our lineage, the evolutionary line that eventually produced you sitting wherever you are right now, extends back 7 million years. That means recorded human history represents approximately 0.07% of the time our evolutionary line has existed on this planet. Sit with that for a moment. Everything you know about human history, every empire, every war, every invention, every king, every plague, every discovery, all of that happened in the last tiny sliver of a story that began when Africa was tearing itself apart along geological fault lines, when the climate was shifting in ways that destroyed entire ecosystems, when a population of apes living in forests found those forests disappearing beneath them. Those apes did not choose to evolve. They did not decide to become human. They were pushed by climate, by geography, by starvation, by extinction events that killed most of their relatives, and by random mutations in their DNA that most of the time did nothing useful at all, but occasionally, rarely, produced something that helped one individual survive just slightly longer than its neighbors. That is the real origin story of the human species. Not a beginning, a process. And that process is what this documentary is about. We are going to go back 7 million years. We are going to watch step by step as a lineage of creatures changes so slowly that no individual alive at any point in this story would have noticed anything different about its own species. We are going to watch brains grow. We are going to watch bodies change their shape. We are going to watch the first stone tools get made and the first fires get lit, and the first migrations carry our ancestors out of Africa and across the entire planet. We are going to meet the species that lived alongside our direct ancestors, the Neanderthals, who built tools and buried their dead, and whose DNA still lives inside the cells of everyone reading these words with European ancestry. We are going to meet the Denisovans, a species we identified entirely from a single finger bone and a tooth, and whose genetic legacy still shapes the immune systems and physical adaptations of populations living today in Asia and the Pacific Islands. We are going to meet Homo floresiensis, a species of human beings that stood 1 meter tall and lived on an island in Indonesia until possibly as recently as 50,000 years ago. We are going to ask the real question, not just where did humans come from, but what does it even mean to be human, and when exactly did that threshold get crossed? And we are going to answer it, with the science, all of it. No mythology, no poetry, no comfortable stories. Just the actual evidence, the actual dates, the actual DNA, the actual fossils, the actual facts about who and what we are and where we came from. This is the story of the first human. Let's go back to the beginning. Chapter 1. Before the beginning. The planet that made us. To understand where the first human came from, you have to understand the world that made it necessary for the first human to exist at all. And that world was in crisis. About 34 million years ago, a major climate shift began that would, over tens of millions of years, fundamentally transform the African continent. The planet was cooling. The ice sheets at the poles were growing, global temperatures were dropping. And in Africa, this cooling and drying process was destroying the environments that the ancestors of both humans and chimpanzees depended on to survive. To understand what was happening, you need to think about what Africa looked like before this process began. 50 million years ago, a large portion of the African continent was covered in dense, closed-canopy tropical forest. Continuous forest. Not the patchwork of forests, savannas and grasslands you see today, but a vast connected belt of trees stretching across much of the continent. This was the environment in which the ancestors of all modern primates were evolving. Warm, wet, dense with vegetation, full of fruit and insects, and the kind of stable food supply that allowed relatively large-bodied primates to survive. Then, slowly, this began to change. The cooling of the planet was connected to several geological processes. The movement of tectonic plates, the rise of the Himalayas, which altered atmospheric circulation patterns, the opening and closing of ocean gateways that changed how heat moved around the globe. None of these processes happened fast. They happened over millions of years. But their cumulative effect on Africa was dramatic. The forest shrank. As the climate became cooler and drier, the dense tropical forest began fragmenting. Patches of forest became separated by open grassland. Rivers changed course. Lake levels rose and fell, and the creatures living in these environments were forced to either adapt, migrate, or die. This process of forest fragmentation is not just a backdrop to human evolution. It is the engine of human evolution. Here is why. When a forest fragments, When one large connected forest breaks into multiple smaller isolated patches, the populations of animals living in those patches become genetically isolated from each other. They can no longer breed with populations in other patches. They evolve separately. And over thousands of generations, populations that were originally the same species begin to diverge. They develop different physical features, different behaviors, different adaptations to their slightly different local environments. And eventually they become different species. This process, geographic isolation leading to genetic divergence leading to speciation, is one of the primary mechanisms by which new species form. And the repeated fragmentation and reconnection of African forests over the last several million years created the conditions for explosive speciation among the primates living in those forests, including the lineage that would eventually produce us. Now, there is another geological process happening at exactly the same time, and it is equally important to understand. Africa is tearing apart. The East African Rift System, a series of geological fault lines running from the Afar Triangle in Ethiopia south through Kenya, Tanzania and Mozambique, has been actively rifting for the last 30 million years. This rifting process is driven by the separation of tectonic plates. The African plate is slowly splitting, with the Somali plate peeling away from the Nubian plate along these fault lines. And as the plates separate, the land in between drops. Great valleys form. Volcanoes erupt along the fault lines. Mountain ranges rise up on either side of the rift. These mountains matter enormously for understanding human evolution. When mountain ranges rise up to the east of the rift, they block moisture-laden winds coming in from the Indian Ocean. The eastern side of the rift, in the rain shadow of these mountains, becomes drier. The forests on the eastern side of the rift shrink faster than the forests on the western side, and the populations of primates living on the eastern side of the rift are exposed to a more open, drier, more variable environment than their relatives on the western side. This is the East Side Story hypothesis, an idea proposed by the French paleontologist Yves Coppens in the 1980s. And while the full hypothesis has been refined and debated significantly since then, the core observation stands. The environment east of the rift was systematically different from the environment west of the rift. And it is in the eastern environments, the drier, more open environments, that the fossil record of early human relatives is richest. While the great apes, chimpanzees, gorillas, bonobos evolved and survived in the wetter forests to the west. But here is what really matters about this environmental context, and why it is so critical to understanding what came next. Open environments are dangerous. In a dense forest, a primate is surrounded by trees. It can escape predators by climbing. It can find food at multiple levels. Fruit in the canopy, insects and small animals at mid-level, roots and tubers at ground level. It has cover, shade, and relatively stable temperatures. The forest provides a buffer against the full brutality of the environment. But in open grassland, or even in the mixed woodland grassland environments that replace the forests, everything changes. Large carnivores, big cats, hyenas, wild dogs are optimized for open environments. They can see and chase prey across open ground far more effectively than in dense forest. And a mid-sized primate living in these open environments, without the speed of a gazelle, without the armor of a buffalo, without the size and strength of an elephant, is exposed in ways it never was in the forest. So the question becomes, how do you survive in open country when you're a primate? This is the question that human evolution spent the next several million years answering. And the first answer, the first major adaptation to open country life that shows up in our fossil record, is one that seems almost absurdly simple. Stand up. Walk on two legs. Bipedalism, walking on 2 feet instead of 4, is the oldest uniquely human adaptation we can document in the fossil record. It appears millions of years before big brains, millions of years before stone tools, millions of years before any of the cognitive or technological capabilities we associate with being human. The very first thing that set our lineage apart from the other apes was not intelligence. It was posture. But why? Why would standing upright help you survive in open country? There are several answers to this question, and they are all probably partially correct. First, bipedalism frees the hands. When you walk on 2 legs, your arms are no longer needed for locomotion. They can do other things: carry food, carry infants, carry objects, make and use tools. The freeing of the hands from locomotion opens up an entirely new category of behavioral possibilities. And while the earliest bipeds almost certainly were not using tools in any sophisticated way, the anatomical foundation for tool use was being laid the moment the hands stopped being needed for walking. Second, bipedalism may have been more energetically efficient for long-distance travel in open country. Chimpanzees walking on 4 limbs expend significantly more energy per unit distance than humans walking upright. In an open environment where food is widely dispersed and you have to travel long distances to find it, a locomotion style that costs less energy per kilometre travelled is a significant advantage. Research by the biologist David Reichlen and others has shown that human bipedal walking is actually quite energy efficient, comparable to or better than the quadrupedal locomotion of most mammals of similar body size. Third, being upright puts the head higher. In tall grass, a bipedal primate can see over the vegetation. It can spot predators at greater distance. It can monitor a wider area. The sensory advantage of height in open country should not be underestimated. Fourth, and this is a hypothesis proposed by Pete Wheeler in the 1990s, being upright may have helped with thermoregulation. In equatorial Africa, the midday sun is intense. A quadrupedal animal presents a large horizontal surface area to direct overhead sunlight. A bipedal animal, standing upright, presents a much smaller surface area to direct overhead sun, while presenting larger surface areas to cooling breezes. And because our early ancestors were losing their fur at approximately the same time, evolving the sweating-based cooling system that is unique to humans among the primates, the combination of upright posture and sweating would have allowed our ancestors to remain active during the hottest part of the day, when most predators and competitors were resting. None of these advantages individually might be enough to drive the evolution of bipedalism on its own. But together, the freed hands, the energy efficiency, the improved visibility, the thermoregulatory benefits, they create a package of advantages in open country that was apparently sufficient for natural selection to favor individuals who walked more upright over individuals who walked less upright, over millions of generations. But here is the thing about bipedalism that nobody tells you in most documentaries about human evolution. It is not free. Walking on two legs imposes costs on the body that are severe, specific, and completely unique to the human lineage. And those costs have defined enormous amounts of human biology, human medicine, and human social structure in ways we are still living with today. When you redesign a skeleton that evolved for four-limbed locomotion into a skeleton that walks on two limbs, you create structural problems. The spine now bears the full weight of the upper body vertically, rather than distributing it horizontally along a horizontal backbone. This is why lower back pain is essentially universal in adult humans, and essentially absent in other apes. Our spines were not designed from scratch for bipedalism. They were modified from a quadrupedal design, and the modifications are imperfect. The foot is completely restructured. The big toe loses its gripping ability and becomes aligned with the other toes. The arch develops to absorb impact. The heel enlarges to bear weight. This is a remarkable engineering solution, but it means the human foot is no longer a grasping organ. Our relatives, the chimpanzees, can grip branches with their feet almost as effectively as with their hands. We cannot. We traded gripping feet for walking feet, and that trade is irreversible. And then there is the pelvis. And here is where we need to spend some time, because the pelvis is where bipedalism created one of the most profound constraints in all of vertebrate biology. To walk efficiently upright, the pelvis needs to be shaped in a specific way, relatively narrow and vertical, to position the hip joints properly beneath the body's center of gravity. But to give birth to large-brained infants, the pelvis needs a large birth canal, a wide, round opening through which the infant's head can pass. These two requirements are in direct conflict with each other. A narrower pelvis is better for walking. A wider pelvis is better for giving birth. And as the brain of our lineage began to grow over millions of years, as the skull of infant Homo sapiens became larger and larger, this conflict became more and more acute. This is called the obstetric dilemma, and it is real, and it is severe, and it has shaped human biology in profound ways. Human childbirth is dramatically more dangerous and more painful than childbirth in any other ape. In chimpanzees, birth is typically fast. The infant can emerge facing forward, and the mother often delivers alone or with minimal assistance. In humans, birth is slow. The infant must rotate during delivery, and without assistance, the risk of maternal and infant death is substantial. Human birth is so difficult precisely because the brain grew faster than the pelvis could widen without destroying our ability to walk. The solution evolution found to this dilemma is remarkable and has profound social implications. Human infants are born premature by the standards of other primates. A chimpanzee infant is born neurologically much more mature than a human infant. It can cling to its mother almost immediately. It can feed independently much sooner. A human infant is completely helpless for months after birth. It cannot walk, cannot feed itself, cannot regulate its own temperature. A human infant requires continuous intensive care for at least the first year of life, and significant support for years beyond that. But this prematurity is the solution to the obstetric dilemma. By being born before the brain fully develops, the infant's skull is still small enough to pass through the birth canal. The massive brain growth that makes humans cognitively unique happens after birth, outside the womb, where it is not constrained by the dimensions of the maternal pelvis. Approximately 90% of human brain growth happens after birth. No other primate comes close to this level of postnatal brain development. And this means something absolutely critical about human social structure. A human infant that is completely helpless for months after birth, that requires years of intensive care, cannot be raised by a single individual. The metabolic demands on the lactating mother are already extreme. Adding full-time infant care to full-time foraging for food is physiologically impossible for one person. Human infants require allomaternal care, care provided by individuals other than the mother. Fathers, grandmothers, older siblings, other group members. The human social group, the extended family, the community, the village, the tribe, may have its deepest roots not in culture or language or even cooperation for hunting, but in the simple biological necessity of caring for infants whose brains grew so large that they had to be born before they were ready. Bipedalism, in other words, began a cascade of changes that ultimately produced not just a different body shape, but a different social system, a different childhood, a different relationship between generations, and a different set of dependencies between individuals. All of this from the decision, or rather from the natural selection that favoured standing up. But we're getting ahead of ourselves, because before we get to big brains and social complexity and all of that, we need to go back to the actual beginning. We need to go back to the moment, as best as the fossil record can show us, when our lineage first separated from the apes. And that moment, as we are about to see, is much older, much stranger, and much more ambiguous than most people realize. Chapter 2. The Split. The moment our line separated from the apes. The most reliable tool we have for figuring out when the human lineage diverged from the chimpanzee lineage is not the fossil record. It is the genome. Every time a cell divides, the DNA in that cell gets copied. And every time the DNA gets copied, there is a small probability of an error, a mutation. Most mutations are neutral. They do not help or harm the organism. They are just random changes in the sequence of DNA bases. And these neutral mutations accumulate at a roughly constant rate over time, a rate we can measure by comparing the genomes of living species whose divergence time we know from other evidence. This is the molecular clock. By counting the number of neutral DNA differences between two species, say between humans and chimpanzees, and dividing by the known rate at which those differences accumulate, you can estimate how long ago those two species shared a common ancestor. When scientists applied the molecular clock to the human-chimpanzee split, the answer they got was startling. Humans and chimpanzees diverged somewhere between 5 and 8 million years ago. The most commonly cited estimate is approximately 6 to 7 million years ago. Think about what that means. The genetic difference between you and a chimpanzee has been accumulating for less than 8 million years. You and a chimpanzee share approximately 98.7% of your protein-coding DNA sequence. The 1.3% difference is what accounts for everything. The upright posture, the large brain, language, technology, civilization, art, mathematics, science. 1.3%. But even that number undersells the similarity, because DNA sequence similarity is only one way to measure genetic relatedness. The more you look at the structural and functional similarities between human and chimpanzee biology, the immune system, the nervous system, the basic biochemistry, the more the differences look like minor modifications of a shared underlying design. Now, the molecular clock gives us a date range. But what does the fossil record tell us about the actual creatures living at the time of this divergence? This is where things get complicated and fascinating. The oldest known hominin fossil, that is, a fossil belonging to the human side of the human-chimpanzee split, is called Sahelanthropus chadensis, and it was found not in East Africa, where most early hominin fossils have been found, but in Chad, in Central West Africa. It was found in 2001 by a team led by the French paleontologist Michel Brunet. Here is what makes Sahelanthropus extraordinary. It is dated to approximately 7 million years ago, right at, or possibly even slightly before, the estimated time of the human-chimpanzee split. And the skull, while in many ways ape-like in its small brain and large brow ridges, has a feature that is, or appears to be, associated with upright locomotion. The foramen magnum, the hole in the base of the skull through which the spinal cord passes, in Sahelanthropus appears to be positioned toward the center of the base of the skull, rather than toward the back. This is significant because the position of the foramen magnum tells you something about the posture of the animal. In quadrupedal animals, where the spine is roughly horizontal, the foramen magnum is at the back of the skull, angled downward and backward, so the spinal cord connects to the base of the skull at an angle appropriate for a horizontal spine. In bipedal animals, where the spine is vertical, the foramen magnum is at the bottom center of the skull, so the spinal cord goes straight up into a skull that is balanced on top of a vertical spine. If Sahelanthropus chadensis genuinely had a foramen magnum positioned toward the center of the skull base, then this 7-million-year-old creature may have been walking, or at least partially walking, upright. That would push the origin of bipedalism back to almost exactly the time of the human-chimpanzee split. But there is a major caveat here, and it is important. We have almost all of the skull of Sahelanthropus. But we do not have postcranial skeleton. We do not have leg bones or hip bones or vertebrae that would directly tell us how this creature moved. The inference about bipedalism is based entirely on the skull morphology, and particularly on the foramen magnum position. And this inference has been contested. In 2020, a paper was published by a team including the French researchers Frank Guy and others, examining a femur bone found at the same site as the Sahelanthropus skull. This femur, if it belongs to Sahelanthropus, shows features that some researchers argue are more consistent with climbing behavior than with bipedal walking. This analysis is disputed, and the debate is ongoing. What this tells us is something important about the nature of early hominin locomotion. It may not have been a clean transition from quadrupedalism to bipedalism. The early stages of our lineage may have included creatures that spent significant time climbing in trees as well as moving bipedally on the ground. Creatures that were neither committed quadrupeds like the apes, nor fully committed bipeds like modern humans, but something in between. Experimenters, adapters, animals whose bodies could do both things imperfectly. Now, approximately 4.5 million years ago, two things become somewhat clearer in the fossil record. We start to find fossils that are unambiguously on the human side of the split, and that show increasing evidence of bipedalism. The genus is Ardipithecus. Ardipithecus ramidus, found in Ethiopia and dated to approximately 4.4 million years ago, was a creature of extraordinary scientific importance when the full analysis was published in 2009. Its remains, informally called Ardi, include enough of the skeleton to reconstruct how it moved. And what the anatomy tells us is fascinating. Ardi was not a chimpanzee-like knuckle walker. Modern chimpanzees walk on the ground by placing their weight on their knuckles, a locomotion style called knuckle walking. Ardi's anatomy, particularly the wrist and hand bones, does not show the modifications associated with knuckle walking. This suggests that knuckle walking in chimpanzees evolved after the human-chimpanzee split, rather than being the ancestral locomotion from which human bipedalism developed. Our common ancestor with chimpanzees may never have walked on its knuckles. At the same time, Ardi's foot has a feature that is decidedly ape-like. The big toe is divergent, sticking out to the side, allowing the foot to grasp branches. Ardi was apparently still spending time in trees, still needing a grasping foot for climbing. The locomotion reconstructed for Ardi is something like cautious bipedalism on the ground, combined with careful above-branch walking in trees, a creature transitioning between worlds, committed to neither forest nor open ground. The brain of Ardipithecus was small, approximately 350 cubic centimeters, comparable to a modern chimpanzee. There is no evidence of tool use, no evidence of complex social behavior beyond what we might expect of an ape-like creature. So, at 4.5 million years ago, what we have on our lineage is a creature that walks bipedally, at least on the ground, but still spends significant time in trees, has a brain the size of an ape's, and lives in a habitat that was probably a mosaic of forest and open woodland, It is on the human side of the family tree. But it is not human in any meaningful sense beyond that. And now the story gets considerably more interesting. Because between about 4 million and about 2 million years ago, the human lineage underwent a remarkable diversification. Multiple species evolved, competed, and in most cases went extinct. The fossil record of this period is rich enough to show us real complexity. Multiple kinds of bipedal apes, slightly different in anatomy, probably occupying slightly different ecological niches. This is the era of Australopithecus. Chapter 3. The walking experiment, Australopithecus and why bipedalism changed everything. On the 30th of November 1974, a team of paleoanthropologists led by Donald Johanson was surveying a dry stream bed in the Afar region of Ethiopia when one of the team members spotted a small fragment of bone sticking out of the sediment. That fragment turned out to be the elbow bone of an individual who had died approximately 3.2 million years ago. Over the next 3 weeks, the team excavated the site and recovered approximately 40% of a skeleton, the most complete early hominin skeleton ever found at that time. The team celebrated that night, and someone played the Beatles song "Lucy in the Sky" with diamonds on a tape player. The skeleton was female. She was small, approximately 1.1 meters tall and 29 kilograms. She was given the name Lucy. Her species designation was Australopithecus afarensis. Lucy changed our understanding of human evolution in ways that are difficult to overstate. Before Lucy, there was a debate in paleoanthropology about which came first in human evolution. The big brain, or bipedalism. Did our ancestors walk upright because they were already smart? Or did they become smart after they started walking? Lucy settled this debate. Her brain was tiny, approximately 410 cubic centimeters, about the same size as a modern chimpanzee's brain, and roughly 1/3 the size of a modern human brain. But her skeleton showed clear adaptations to bipedal walking. The shape of her pelvis, the angle of her thigh bones, the structure of her knee joints and ankle joints. All of it consistent with an animal that walked upright on 2 legs as its primary means of locomotion. Bipedalism came first. The brain came later, much later. Now, to be clear, Lucy's bipedalism was not identical to modern human bipedalism. Her arms were relatively long compared to her legs. Her fingers were curved, suggesting she still climbed trees regularly. Her hip structure, while modified for bipedalism, retained some ape-like features. She probably walked with a slightly different gait than modern humans. Some researchers have argued for a slightly wider stance, a different pelvic tilt. But she walked on 2 feet, and she had done so for her entire life. Her species, Australopithecus afarensis, is currently the oldest hominin species for which we have clear, unambiguous evidence of bipedalism from both skeletal anatomy and extraordinarily fossilized footprints. In 1978, at the site of Letoli in Tanzania, a team led by Mary Leakey discovered something extraordinary preserved in volcanic ash. Footprints. Fossilized footprints dated to 3.6 million years ago, made by at least 2 individuals, and possibly 3, walking upright across a freshly deposited layer of volcanic ash that subsequently hardened like concrete. The prints extend for approximately 27 meters. The individuals who made them are gone. Their bones are gone. But their footprints remain, perfectly preserved, as tangible a proof of bipedal locomotion as you could possibly ask for. Those footprints are among the most moving objects in all of paleoanthropology. 2 beings, 3.6 million years ago, walking side by side across a landscape being rained with volcanic ash from an eruption in the distance. They left no tools, no art, no language, just footprints. And those footprints look like the footprints of a human being. Australopithecus afarensis survived as a species for approximately 900,000 years, from approximately 3.9 million years ago to approximately 3 million years ago. That is nearly 1 million years. For perspective, our own species, Homo sapiens, has existed for somewhere between 300,000 and 315,000 years. Australopithecus afarensis survived 3 times longer than we have, but its brain never grew significantly, and as far as we can determine, it never made stone tools. It was a successful, persistent, long-lasting species that occupied the open woodlands and grasslands of East and maybe Central Africa for nearly 1 million years. But by the cognitive standards we use to define humanity, it was not human. It was an upright walking ape. Now, the genus Australopithecus was not a single species. It was a radiation of species, multiple species, evolving from earlier Australopithecine ancestors, adapting to slightly different environments, developing slightly different body plans and dietary specializations. The fossil record from this period, roughly 4 million to 2 million years ago, shows us a world with at least 6 or 7 distinct hominin species existing across Africa. There is Australopithecus afarensis, Lucy's species. There is Australopithecus africanus, found in South Africa slightly later in time, with a slightly rounder skull. There is Australopithecus sediba, found in South Africa and dated to approximately 2 million years ago, with an intriguing mix of features that some researchers argue show it transitioning toward the genus Homo. There are the robust Australopiths, sometimes placed in their own genus, called Paranthropus, with massive jaws and huge molars, specialized for eating hard, tough foods like seeds and tubers. Paranthropus boisei, sometimes called Nutcracker Man for its enormous jaw and cheek teeth. Paranthropus robustus in South Africa. All of these species coexisted, at least in different regions or at different times. All of them were bipedal. All of them had small brains by modern human standards. All of them eventually went extinct, most of them without leaving any descendants. The robust Australopiths are perhaps the most instructive. Paranthropus species evolved massive reinforced chewing apparatus, thick enamel, huge molars, a sagittal crest on the top of the skull, a bony ridge to which massive jaw muscles attached. They were clearly specialized for a hard food diet. And this specialization worked. They survived for well over 1 million years. but they were an evolutionary dead end in terms of the lineage that leads to us. They were specialists, and specialization, while useful in stable environments, is risky when environments change drastically. Meanwhile, somewhere in this complex of species, from roughly 2.8 million to 2 million years ago, something different was happening. A lineage within the Australopithecines was beginning to show something new. Their brains were getting bigger, and they were making tools. The earliest stone tools in the archaeological record date to approximately 3.3 million years ago, found at a site called Lomekwi 3 in Kenya. These tools, called Lomekwian tools, are simple. Rocks that have been deliberately struck to create sharp edges. They are crude, but they are intentional. They are unambiguous evidence that some hominin 3.3 million years ago understood that you could use one rock to strike another rock in a specific way to create a sharp cutting edge. But here is the thing. 3.3 million years ago, the only hominins we know of in East Africa are Australopiths. The first species we traditionally assign to the genus Homo, Homo habilis, does not appear in the fossil record until approximately 2.4 million years ago. This means either Australopiths were making stone tools, which would require us to significantly revise our understanding of their cognitive capabilities, or there was a species of early Homo existing at 3.3 million years ago that we have not yet found fossil evidence for. Or both. The honest answer is, we do not yet know who made the Lomekwi tools. The field is actively debating this question, and it illustrates something important about paleoanthropology as a science. It is not a completed field with all the answers. It is an active science where major discoveries and revisions happen regularly, where a single new fossil or a single new dating result can overturn assumptions that have been held for decades. What we do know is that by approximately 2.6 million years ago, a more standardized and widespread stone tool tradition appears in the archaeological record, called the Oldowan, named after Olduvai Gorge in Tanzania, where many early examples were found. Oldowan tools are simple stone flakes and the cores they were struck from. Simple but effective. Experimental studies have shown that Oldowan flakes are sharp enough to butcher animal carcasses and cut through hide and tendons that primate teeth could not manage. And these Oldowan tools are associated with the first members of our own genus, the genus Homo. Chapter 4. The Brain Explosion. How Homo habilis crossed the threshold. Something changed around 2.8 to 2.4 million years ago. The fossil record shows a hominin skull that is different from the Australopiths, not radically different, not immediately dramatically different in every respect, but different in ways that are significant. The brain is bigger, the face is slightly flatter, the teeth are slightly smaller, the skull is rounder, and this creature appears to be making tools with greater consistency and sophistication than anything that came before. This is Homo habilis, the first member of our own genus. Discovered in the 1960s at Olduvai Gorge in Tanzania by Lewis and Mary Leakey's team. Named Homo habilis, handyman, because of its association with stone tools. The brain of Homo habilis was approximately 600 to 700 cubic centimeters. Compare that to the 400 to 500 cubic centimeters typical of Australopithecus and the approximately 1300 to 1400 cubic centimeters of a modern Homo sapiens. Homo habilis was about halfway between the Australopiths and us in raw brain volume. But raw brain volume is not the whole story. Brain organization matters as much as size. There is an area of the brain called Broca's area, a region in the left frontal lobe that is involved in the production of language. In living humans, this region is enlarged relative to other primates. Endocasts, casts of the interior of fossil skulls, which preserve the general shape of the brain, suggest that in Homo habilis this region may already have been slightly enlarged relative to the Australopiths. Some researchers have interpreted this as early evidence for the neural substrates of language beginning to develop in Homo habilis. Others are more cautious. The evidence is indirect and debated. But here is the more fundamental question. Why did the brain start growing at this particular time? The answer appears to be connected to a combination of factors, and the most important of them is diet. About 2.6 million years ago, there is a major cooling and drying event in Africa. The climate becomes more variable and more extreme. Forests contract further. Open grasslands expand. And the dietary options available to a mid-sized primate in these open environments change significantly. One of the major dietary shifts that appears to have occurred in early Homo, documented through stable isotope analysis of fossil teeth, is a significant increase in the consumption of animal protein. Meat, and specifically the marrow inside bones. Here is why this matters for brain growth. The brain is the most metabolically expensive organ in the body. It represents approximately 2% of the body weight of a modern human adult, but it consumes approximately 20% of the body's energy budget. Feeding a large brain requires a high-quality, calorie-dense diet, and meat, with its high protein content and high fat content, is a significantly more calorie-dense food source than the plant foods that made up the bulk of the Australopith diet. The hypothesis is essentially this. As early Homo began accessing more animal protein, whether through active hunting, through scavenging the kills of large predators, or through early forms of cooperative food acquisition, the increased energy availability made it possible to sustain a larger brain. And a larger brain, in turn, made it possible to develop better strategies for acquiring food, which provided even more energy to sustain an even larger brain. A positive feedback loop between diet, energy availability, and brain size. The tools were critical to this process. An Aldebaran stone flake is not just a tool, it is a metabolic key. A large animal carcass, a zebra, an antelope, even a large elephant that died of natural causes, contains enormous amounts of calories. The meat on the outside can be eaten by carnivores with the appropriate teeth, but the bones contain marrow, fat-rich, calorie-dense marrow, that most carnivores cannot access. A hyena can crush bones. But most of the large predators of the African savanna cannot easily access bone marrow. A hominin with a sharp stone flake can. By striking bones with stone tools to break them open, early Homo could access a calorie source that was largely unavailable to competitors. This is called a fallback food, a resource available when primary food sources are scarce, and one that requires a specific technology to access. The stone tool was the technology that unlocked this calorie source, and unlocking this calorie source provided the energy to sustain a larger brain. But there is another dimension to early brain growth that is equally important, and it is social. Big brains are not just for manipulating the environment. They are for navigating complex social relationships. The social brain hypothesis, proposed by the British anthropologist Robin Dunbar, argues that the primary selective pressure for large brains in primates, including humans, was the cognitive demand of living in large, complex social groups, tracking who is friends with whom, remembering who helped you in the past and who cheated you, managing alliances and coalitions, predicting the behavior of other individuals, understanding the mental states of others, what researchers call theory of mind. These are cognitively demanding tasks, and the larger and more complex the social group, the more demanding these tasks become. Dunbar showed a statistical relationship across primates between the size of the neocortex, the outermost, most recently evolved layer of the mammalian brain, and the typical social group size of the species. Species with larger neocortices tend to live in larger social groups, and humans, with the largest neocortex relative to brain size of any primate, live in extraordinarily large and complex social groups. The social demands of group living and the energetic demands of a high-quality diet probably both contributed to the brain expansion that began with early Homo. They are not competing explanations. They are complementary pressures, both pushing in the same direction, both contributing to the same outcome. Now, Homo habilis is a contentious species, and it is worth acknowledging that contention directly. When Louis Leakey and his colleagues proposed the species in 1964, they were arguing that it represented the first true member of the genus Homo, the first creature that was, in some meaningful sense, human. But subsequent fossil discoveries have complicated this picture considerably. The fossils assigned to Homo habilis show considerable variation. So much variation that some researchers have argued the category actually includes multiple species. Some have proposed splitting the habilis specimens into at least two species, Homo habilis itself and a separate species called Homo rudolfensis, which has a larger brain and somewhat different facial proportions. Others maintain that this variation falls within the range of a single species. The debate is ongoing. What is not debated is that by approximately 2 million years ago, there are hominins in the fossil record with significantly larger brains than the australopiths, associated with stone tool technology, showing a suite of anatomical features that are recognizably more human than anything that came before. Whether you call all of them Homo habilis, or split them into two species, or refine the taxonomy in some other way, the pattern is clear. Brain size was increasing, complexity was increasing. The trajectory towards something we might recognize as human was underway. But Homo habilis never left Africa. Its body was still small. In some specimens, not much larger than a robust Australopith. Its legs were still relatively short compared to its arms, suggesting it may have spent significant time in trees. It was, in the terminology of the field, a transitional species, clearly more derived than the Australopiths, clearly less derived than what would come next. What came next was something genuinely different, something with a body built for endurance, something with a brain that had crossed a qualitative threshold, something that, for the first time in the history of this lineage, had the physical and cognitive capacity to do what no hominin before it had ever done. Leave Africa. Chapter 5. Out of Africa, the first time. Homo erectus conquers the world. In 1984, at a site called Nariokotome on the western shore of Lake Turkana in Kenya, a paleoanthropologist named Kamoya Kaimu found a small piece of skull lying on the surface of the ground. It belonged to a boy, approximately 8 years old, and approximately 1.6 million years old. Over several seasons of excavation, the team recovered nearly the entire skeleton of this individual, the most complete skeleton of an early Homo ever found, surpassing even Lucy in its completeness. He was given the informal name Turkana Boy. His species was Homo erectus. Turkana Boy was a revelation. He was tall. Projections based on his age and skeletal development suggest he would have reached approximately 1.8 meters as an adult. His legs were long relative to his arms, the modern human body proportion, optimized for long-distance bipedal locomotion rather than the more ape-like proportions of the Australopiths and Homo habilis. His brain was approximately 900 cubic centimeters, larger than Homo habilis, not yet as large as modern humans. His nose projected forward, as does the modern human nose, rather than being flat like an ape's. An adaptation possibly related to humidifying and warming air before it reaches the lungs during extended aerobic exercise. Homo erectus, or its immediate African predecessor, which some researchers call Homo ergaster, though many treat the two as variants of the same species, represents a genuine threshold in human evolution. Not just a quantitative improvement on what came before, but a qualitative leap. The body of Homo erectus is built for endurance running. The long legs, the arched foot, the nuchal ligament, a ligament at the back of the neck that stabilizes the head during running and is absent in other apes. The waist and the rotational capacity of the torso, the external nose, the relatively hairless skin with its dense sweat glands. All of these features are interpreted as adaptations to long-distance pursuit predation. The hypothesis, proposed by Dennis Bramble and Daniel Lieberman in 2004, is that Homo erectus, or its immediate ancestors, developed the ability to run long distances as a primary hunting strategy. Not to sprint like a cheetah, but to pursue prey at a moderate pace over many kilometers, using the capacity for sweating-based thermoregulation to outlast prey animals that thermoregulate through panting and cannot pant effectively while running at speed. This is called persistence hunting. and it has been documented in some traditional human hunter-gatherer groups in Africa and elsewhere. A group of hunters tracks a prey animal, typically during the hottest part of the day, when the prey is at greatest risk of heat stress, and runs it down over many kilometers until the animal overheats and can no longer move. The tools of Homo erectus, the Acheulean hand axe tradition, which appears at approximately 1.7 million years ago, also represent a significant advance over the older one. The Acheulean hand axe is a teardrop-shaped stone tool that is bifacially flaked, worked on both sides, to produce a symmetrical standardized form with a sharp cutting edge running most of its perimeter. To produce an Acheulean hand axe, you need to plan ahead, to hold in your mind an image of the finished product and work systematically toward it, removing flakes in a specific sequence. This requires a level of planning and mental projection that is cognitively more demanding than the opportunistic flaking of older one tools. And Homo erectus made these tools in enormous quantities across a geographic range that spanned Africa, Europe, and Asia for approximately 1.5 million years. Which brings us to the most significant thing about Homo erectus. It left Africa. The earliest evidence of hominins outside Africa comes from a site called Dmanisi in the country of Georgia, dated to approximately 1.8 million years ago. The fossils found at Dmanisi are now attributed to Homo erectus, and they represent the oldest known hominin remains outside of Africa. 1.8 million years ago, these individuals, several of them representing adults and possibly juveniles, had walked or been born in a location that was thousands of kilometers from the African sites where similar fossils have been found. How did they get there? What were they doing? Did they migrate deliberately, following game herds? Did they expand gradually, generation by generation, never moving more than a few dozen kilometers from their birth location in any single lifetime, but accumulating distance over thousands of generations? We do not know. We cannot ask them. But the fact that they were there, in Georgia, 1.8 million years ago, is unambiguous. From Georgia, or from Africa, or from multiple dispersal events, Homo erectus spread across the Old World. Fossils and tools attributed to Homo erectus have been found in Java in Indonesia. The original Java man specimens, discovered in the 1890s and dated to between 1.5 million and 500,000 years ago. In China, the Peking man specimens from Zhoukoudian, dated to between 700,000 and 200,000 years ago. In Europe, though the European record is complicated, with different researchers attributing the earliest European hominins to Homo erectus, or to a related species called Homo antecessor, or to Homo heidelbergensis. Homo erectus was the most geographically successful and temporally persistent hominin species that has ever existed. It survived in one form or another for approximately 1.8 to 2 million years. Its fossils span from Africa to Western Europe to Southeast Asia to China. And somewhere in this vast geographic range and this vast span of time, it discovered, or developed, something that would fundamentally change the relationship between our lineage and the rest of the living world. Fire. The controlled use of fire is one of the most transformative technological developments in the history of our species, and the evidence for when it began is a matter of ongoing research and debate. The earliest evidence for fire use, burned bones and hearth structures, at a site called Wonderwerk Cave in South Africa, dates to approximately 1 million years ago. Evidence from Gesher Benot Yaakov in Israel is dated to approximately 790,000 years ago. Both sites are associated with Homo erectus. Some researchers have argued for fire use even earlier, back to 1.5 million years ago. But the evidence is more ambiguous. The implications of fire control are staggering, and most people, when they think about fire, focus on the obvious ones: warmth, light, protection from predators. These are real and important, but they are not the most profound consequence of fire use. The most profound consequence is cooking. The biological anthropologist Richard Wrangham has argued, in a hypothesis he calls the cooking hypothesis, that the development of cooking was the key dietary shift that drove the dramatic brain expansion and body changes visible in the transition to Homo erectus and beyond. Here is the core of the argument. Cooking fundamentally changes the nutritional value of food. It breaks down the cell walls of plant foods, making the starches inside accessible. It denatures proteins in meat, making them easier to digest. It kills pathogens. It softens food, reducing the time and energy required for chewing. The energetic return from cooked food is substantially higher than from raw food. Studies with humans show that people on raw food diets, eating the same number of calories as people eating cooked food, consistently lose weight, because more of the raw food passes through the digestive system undigested. The gut cannot extract as much energy from raw food as from cooked food. Wrangham argues that the fossil evidence supports the cooking hypothesis. At the transition from Australopiths to early Homo, and especially at the transition to Homo erectus, there are changes in the skeleton that suggest a significant reduction in the size of the digestive system. Smaller gut, smaller teeth, smaller jaw muscles, reduced gut capacity, at exactly the time when brain size was increasing. This seems counterintuitive if diet was improving. Normally, a better diet would be accompanied by a larger digestive system. But if the diet improved specifically because of cooking, because cooking pre-digested the food outside the body, reducing the digestive work required, then a smaller gut makes sense. The digestive system could shrink because cooking was doing part of its job. The energy liberated by a smaller gut and a more digestible diet could be redirected to the brain. Wrangham estimates that cooking may have provided enough additional energy to account for a substantial portion of the brain increase seen from Homo habilis to Homo erectus. The cooking hypothesis is not universally accepted. Some researchers argue that the earliest evidence for fire use is too late to explain the early Homo erectus morphological changes, and that the cooking hypothesis requires evidence of fire use earlier than what we currently have. Wrangham and others argue that the absence of early fire evidence may reflect preservation bias. Organic fires on open savanna leave little lasting trace, rather than the absence of fire itself. The debate continues. Whether or not cooking was the critical trigger, what is clear is that Homo erectus was doing something that generated enough energy to sustain a significantly larger brain, build a significantly larger body, and sustain a lifestyle that included long-distance migration across half the known world. It was, in every meaningful physical sense, something close to human. But it is not us. Not yet. And here is where the story becomes genuinely strange. Because while Homo erectus was spreading across the Old World and living in Asia for millions of years, back in Africa, and then gradually spreading into Europe and back into parts of Asia, something else was happening. The lineage was continuing to change, continuing to evolve, and not in isolation. The picture that emerges from the fossil record of the last 500,000 years is not a simple linear progression. Homo erectus begat Homo heidelbergensis, begat Neanderthals and modern humans. The picture is a tangled web of interacting populations, partially separated by geography, evolving in parallel and semi-independently, periodically coming into contact and, as we now know from ancient DNA, interbreeding. The world, for most of the last 500,000 years, was not a world with one kind of human being in it. It was a world with several. Chapter 6. The Crowded World. Multiple species of humans living simultaneously. In 2010, the journal Nature published a paper that permanently changed what scientists knew about the relationship between Homo sapiens and the other human species it had lived alongside. The paper announced that scientists, led by Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology in Leipzig, had successfully sequenced a draft genome from Neanderthal fossils. And the genome revealed something that, until that moment, had been merely speculated. Modern humans with ancestry outside of Africa, Europeans, Asians, the indigenous peoples of the Americas and the Pacific, carry between 1% and 4% of their genome from Neanderthal ancestors. Homo sapiens and Neanderthals had sex. They had children. And those children's descendants are alive today. To understand the full significance of this discovery, we need to understand who the Neanderthals were. Neanderthals, Homo neanderthalensis, were not the brutish, dim-witted cave people of popular imagination. They were a sophisticated, adaptable, cognitively complex species of human being that lived in Europe and Western Asia for approximately 400,000 years. They survived multiple glacial cycles, periods when much of northern Europe was covered by ice sheets, when temperatures were dramatically lower than today, when the fauna of Europe included woolly mammoths, woolly rhinoceroses, cave bears, and cave lions. And they did this without modern technology, without agriculture, without any of the civilizational infrastructure we take for granted. Neanderthal brains were, on average, slightly larger than modern human brains. Approximately 1,400 to 1,600 cubic centimeters. They made sophisticated stone tools, the Mousterian tool tradition, that required planning and multiple manufacturing steps. They hunted large game. Mammoth bones with cut marks from stone tools have been found at Neanderthal sites, and the injuries documented on Neanderthal skeletons are consistent with close contact hunting of large, dangerous animals. They treated their injured. Neanderthal skeletons show healed injuries that could not have healed without extended care from other group members. Individuals who were too injured to feed themselves were kept alive by their group for months or years. They appear to have buried their dead. Whether this burial behavior was ritualistic, whether it reflected any form of belief about death and what comes after, is debated. But the physical practice of intentionally placing bodies in the ground, sometimes with other objects, is documented at multiple Neanderthal sites. There is evidence, disputed but persistent, that Neanderthals may have engaged in symbolic behavior. Shells pierced for wearing as ornaments have been found at Neanderthal sites. Pigments, ochre and other colorants have been found at Neanderthal sites. A cave in Spain called La Pasiega contains markings that have been dated using uranium-thorium dating of calcite overlying them to at least 65,000 years ago, well before Homo sapiens arrived in Europe. If these dates are correct, Neanderthals were making abstract markings on cave walls. None of this makes Neanderthals identical to us. The complexity and diversity of their symbolic and artistic expression appears to be more limited than that of the Upper Paleolithic Homo sapiens, who eventually replaced them. But the picture of Neanderthals as essentially subhuman, as cognitively comparable to Australopiths, as creatures fundamentally different in kind from us, this picture is simply wrong. It is a product of the 19th century cultural moment in which Neanderthal fossils were first discovered and interpreted, and it has been thoroughly demolished by the accumulated evidence of the last several decades. Neanderthals were humans, different from us, probably cognitively different from us in meaningful ways, but humans, and we had sex with them. Now, the Neanderthals are only the most famous of the multiple human species that coexisted with our own ancestors during the crucial period of our species' emergence. The last few decades of research have revealed a world far more populated with human diversity than anyone suspected. In 2010, the same year the Neanderthal genome was published, another paper in Nature announced the discovery of an entirely new species of human being, identified not from a complete skeleton, not from a well-preserved skull, but from the genome sequenced from a single finger bone found in Denisova Cave in the Altai Mountains of Siberia. This species is called the Denisovans. We still do not have a full skeleton of a Denisovan. We have a finger bone, a few teeth, and some bone fragments. We have essentially no idea what a Denisovan looked like. We know from the genome that they were related to Neanderthals. The Denisovan and Neanderthal lineages diverged from a common ancestor approximately 300,000 to 400,000 years ago. We know they lived in Siberia at least during periods covered by the fossil record. We know from ancient DNA found at other sites that they ranged at least as far as Tibet. And we know that modern humans carry Denisovan DNA. Not all modern humans, but many. Modern populations from Melanesia and Papua New Guinea carry 4 to 6% Denisovan ancestry. Aboriginal Australians carry Denisovan DNA. Populations in the Philippines carry Denisovan DNA. Some populations in East and South Asia carry traces of Denisovan ancestry. The high-altitude adaptation gene called EPAS1, which allows Tibetan populations to live and work at altitudes that would incapacitate most humans, has been traced to a Denisovan version of the gene that entered the Tibetan gene pool through interbreeding. Without Denisovan DNA, the Tibetan people's extraordinary physiological capacity to function at high altitude might not exist. The Denisovans are, from a genomic perspective, one of the most significant discoveries in the history of paleoanthropology. A species identified entirely from DNA. A species whose physical appearance we cannot describe. A species that interbred with our ancestors and left traces in the genomes of living human populations that still shape their biology today. But the Denisovans and Neanderthals are not the only other human species that existed alongside Homo sapiens. In 2004, a team of Australian and Indonesian researchers published a paper announcing the discovery of fossils from an island in Indonesia, the island of Flores, that represented something extraordinary. These fossils, including a remarkably complete skull and partial skeleton, belonged to an adult individual who stood approximately 1 meter tall and had a brain approximately 420 cubic centimeters in volume. Smaller than Homo habilis. Smaller than some Australopiths. This was not a pathological modern human. This was a separate species, Homo floresiensis, nicknamed the Hobbit, after the small fictional humanoids created by J.R.R. Tolkien. Homo floresiensis appears to have evolved on the island of Flores over hundreds of thousands of years in isolation, experiencing what biologists call insular dwarfism. On islands with limited food resources and no large predators, large-bodied species frequently evolve smaller body sizes over time, because smaller bodies require less food. The fossils from Flores show characteristics consistent with the population derived from an earlier Homo lineage, possibly Homo erectus, possibly something even older, that became stranded on the island and evolved in isolation. Despite its tiny brain, Homo floresiensis appears to have made stone tools, simple but consistent with intentional production, and the fossils are dated to as recently as approximately 50,000 years ago. Some researchers have argued that stone tools at the site extend to as recently as perhaps 12,000 years ago, though the association between those later tools and the Homo floresiensis fossils is uncertain. Think about that. 50,000 years ago, at most 100,000 years ago, there was a species of human being on a Pacific island that stood 1 meter tall and had a brain the size of an Australopith, making stone tools. At the same time that Homo sapiens were crossing the world, creating art, making music and developing complex language, there were other humans, on islands and in remote forests and in mountain caves, living in ways that would have been unrecognizable to modern humans, having evolved in isolation into forms quite different from anything in the main story of human evolution. And there is one more species that needs to be discussed in this context. One that was discovered even more recently, and that has raised some of the most profound questions about what it means to be human. In 2013, in a cave system in South Africa called the Rising Star Cave System, a team of cavers exploring narrow passages deep in the cave found a remarkable sight. Bones. Human-like bones lying on the cave floor in a chamber so deep and so narrow that it required specifically selected slender individuals to crawl through passages barely 20 centimeters wide to reach them. The fossil recovery operation that followed, organized by the paleoanthropologist Lee Berger of the University of the Witwatersrand, retrieved more than 1,500 fossil elements representing at least 15 individuals of all ages, juveniles, adults, elderly. The species they represented was new to science: Homo naledi. Homo naledi is one of the most puzzling hominin species ever discovered. Its anatomy is a mosaic. It has hands and feet that are remarkably human-like, a pelvis and shoulder that are more primitive, and a brain approximately 450 to 560 cubic centimeters in volume, barely larger than Australopithecus. It is not easily placed in the hominin family tree. It shows features associated with very early Homo. Yet it also shows features that are derived, more modern-looking. When it was first described, most researchers assumed Homo naledi was very old, perhaps 2 to 3 million years, based on the primitive features of its anatomy. When radiometric dating was performed and the results published in 2017, the paleoanthropological community was genuinely shocked. Homo naledi was dated to approximately 200,000 to 335,000 years ago. This means Homo naledi was alive at the same time as early Homo sapiens, in the same general region of southern Africa. A small-brained, primitive-looking species of human, living simultaneously with our own ancestors. And Lee Berger's team has argued, controversially, that the pattern of accumulation of fossils in the Rising Star Cave System suggests that Homo naledi was intentionally placing dead bodies deep in the cave. That this small-brained species was practicing something that looks remarkably like mortuary behavior, deliberate disposal of the dead. If true, this would mean that symbolic or ritual behavior associated with death is not uniquely associated with large brains. That the cognitive threshold for this kind of behavior is lower than we assumed. This interpretation is disputed, and the debate is ongoing. But it illustrates the point powerfully. The relationship between brain size and behavioral complexity in the human lineage is not simple, and our understanding of it is still developing. The world of 500,000 to 50,000 years ago, in other words, was a world rich with human diversity. Multiple species, multiple lineages, multiple cognitive and behavioral experiments. And somewhere in this world, in Africa, probably in Africa, the one species that would eventually outsurvive all the others was developing. Our species, Homo sapiens. Chapter 7. The origin point. When and where Homo sapiens actually appeared. For much of the 20th century, the conventional answer to the question of when and where Homo sapiens first appeared was something like this. Around 100,000 to 200,000 years ago in East Africa, probably in the region of Ethiopia or Kenya. That answer is no longer the whole story. In 2017, a paper published in the journal Nature, reporting on fossils from a site called Jebel Irhoud in Morocco, announced that the oldest known fossils of Homo sapiens had been dated to approximately 315,000 years ago. 315,000 years ago, in Morocco, in northwest Africa, not East Africa. The Jebel Irhoud fossils had actually been known since the 1960s, but they had been difficult to date and their species attribution had been uncertain. New dating using a technique called thermoluminescence on burned flint found in the same sedimentary layer as the fossils produced the 315,000-year date. And a re-examination of the fossils using micro-CT scanning, high-resolution three-dimensional X-ray imaging, confirmed that they belonged to Homo sapiens, though an early form of the species with some features, particularly in the shape of the brain case, that differ from modern humans. What does this mean? First, it pushes the origin of our species back significantly earlier than the previous estimates of 100 to 200,000 years. Second, it places the origin, or at least the earliest known evidence of our species, at the western end of Africa, not the eastern end. This is significant because it undermines any simple narrative about Homo sapiens originating in a single small geographic region of East Africa. Third, and most importantly, it has reinforced a model of human origins that paleoanthropologists have been developing over the last decade, one that is called the Pan-African model, or the African multi-regional model. The traditional model of Homo sapiens origins, sometimes called the East African origin or the Garden of Eden model, envisioned a small ancestral population in East Africa giving rise to our species, which then spread across Africa and eventually out of Africa. The Pan-African model is different. It proposes that Homo sapiens did not evolve from a single population in a single geographic location, but that its defining characteristics emerge gradually and asynchronously across multiple populations spread across the African continent. Different populations in different regions develop different aspects of the modern human anatomy and behavioral repertoire at different times. These populations were not permanently isolated. Africa is not a place where populations cannot move. They were connected sometimes by corridors created when the climate allowed, rivers, forests, coastal routes, and when they came into contact, they interbred, sharing genetic material and potentially cultural and behavioral innovations. The modern human species in this model is not the product of a single founding population. It is the product of a continent-wide mixing of related populations over hundreds of thousands of years, with the final modern human package emerging from this long process of divergence, connection, mixing, and co-evolution. This model is better supported by the fossil evidence than the single origin model. Fossils from different parts of Africa at different time periods show different combinations of modern and archaic features, without a clear linear progression from primitive to modern. Early Homo sapiens from Morocco look different from early Homo sapiens from Ethiopia, which look different from early Homo sapiens from South Africa. But all of them show some features that are recognizably sapiens, and some features that are archaic. It is as if the species was assembled from parts, from innovations that arose in different populations at different times and were gradually combined through gene flow and migration into the modern package we see today. And we need to ask, what is the modern package? What exactly characterizes Homo sapiens anatomically, and how different were the earliest members of our species from us? The skull shape of modern Homo sapiens is distinctive. We have a high, domed, globular brain case. The top of the skull is round, not flat. The face is relatively flat and tucked beneath the brain case rather than projecting forward like an ape's face or a Neanderthal's face. The brow ridges, the bony projections above the eye sockets that are prominent in Neanderthals and Homo erectus, are very small or absent in modern humans. The chin is prominent. Homo sapiens is the only hominin species with a true chin, a projection of bone at the front of the lower jaw. The face is relatively small. Early Homo sapiens from 300,000 years ago, the Jebel-Irhoud individuals, have most of these features, but the brain case is somewhat flatter and more elongated than modern skulls. The face is more prominent. The brow ridges are somewhat larger. They look like early members of our species, recognizably related to us, but not quite modern. This difference in skull shape reflects something important about brain organization, not just brain size. The globular shape of the modern human skull is associated with the expansion of specific brain regions, the parietal lobes, which are involved in spatial reasoning, attention, and sensory integration, and the temporal lobes, which are involved in language and social cognition. The evolution of the modern human brain shape is not complete at 300,000 years ago. It appears to reach its final form somewhere between 100,000 and 40,000 years ago, based on analyses of endocranial shape in fossil skulls. This means something fundamental. The bodies of Homo sapiens were assembled before the minds of Homo sapiens were complete. The 315,000-year-old individuals from Jebel-Irhoud were Homo sapiens by skeletal criteria. They had modern faces, modern postcranial anatomy, modern tooth morphology, but their brains, while modern in size, were not yet organized in the fully modern way. And the behavioral evidence supports this conclusion. The archaeological record from early Homo sapiens sites in Africa, while more sophisticated than what we find at Neanderthal sites in Europe, is not yet the explosive, innovative, symbolically rich record of the Upper Paleolithic, the period that begins about 50,000 years ago. Early Homo sapiens made tools. They used pigments. Ochre, which leaves orange-red marks, has been found at numerous African sites dated to over 100,000 years ago. They show evidence of long-distance trade in shells and other materials. There are isolated instances of seemingly symbolic behavior going back over 100,000 years. But this behavior is episodic, inconsistent. It appears at some sites in some periods and not at others. It does not have the consistent, ubiquitous, overwhelming presence that symbolic and technological behavior has in the archaeological record after about 50,000 years ago. Something changes around 50,000 years ago. Something fundamental. Something that transforms the behavioral profile of our species so dramatically that archaeologists call it the behavioral revolution, or the cognitive revolution, or the Upper Paleolithic revolution. And whatever it was, it is the last major threshold in the story of the first human. Chapter 8. The Behavioral Revolution. When we started thinking like humans around 70,000 to 50,000 years ago, the archaeological record does something it has never done before in the entire multi-million-year history of the hominin lineage. It explodes. Art, music, personal ornaments, complex projectile weapons, long-distance trade networks, burials with elaborate grave goods, fishing, nets, boats, storage of food for later use, evidence of planning across multiple seasons, and eventually the occupation of environments that no previous hominin had ever successfully colonized. Australia, the deep interior of cold Eurasia, ultimately the Americas. The Upper Paleolithic Revolution, or the behavioral modernity transition, is one of the most discussed and debated topics in all of paleoanthropology, because the question of what caused it is still not fully resolved. Let us start with what we know happened. The oldest confirmed representational art in the world, actual images that depict specific recognizable subjects, not just abstract marks, comes from cave sites in Indonesia and Europe dated to approximately 45,000 to 65,000 years ago. A painting of a pig on the Indonesian island of Sulawesi has been dated to at least 45,500 years ago. The extraordinary cave paintings of Lascaux in France and Altamira in Spain are somewhat later, approximately 17,000 to 20,000 years ago, but even earlier examples from the same European cave tradition extend back 40,000 years or more. Cave paintings of this quality and sophistication are not the work of creatures that think like animals. They require the ability to plan, to select pigments, prepare surfaces, sketch outlines, fill in color, and produce images that accurately represent the visual appearance of real animals. They require the ability to work by artificial light, in many cases deep within cave systems where no natural light penetrates. They require, at some level, an aesthetic sensibility, a judgment that one representation looks better than another, that an image is worth making and worth preserving. The earliest personal ornaments, shells deliberately pierced for stringing as beads or pendants, are even older. Perforated shells from sites in Morocco, Israel and Algeria have been dated to between 80,000 and 130,000 years ago. Some researchers have argued for shell ornaments as old as 160,000 years. Personal ornaments are significant because they imply something very specific cognitively. They imply that you care about how other people perceive you. They imply that you understand that your appearance communicates information to other people about who you are, your group membership, your status, your identity. They imply a sense of self that extends beyond immediate experience, a self that can be projected, displayed, modified, and communicated symbolically. This is theory of mind, the ability to model other people's mental states, to understand that other minds exist, to think about what others think and perceive and believe. and theory of mind is fundamental to language, to culture, to the complex social dynamics that characterize human societies. The Blombos cave site in South Africa has produced some of the oldest evidence of these kinds of behaviors. From layers dated to approximately 77,000 years ago, archaeologists have recovered perforated tick shells used as beads, engraved pieces of ochre showing deliberate geometric designs, and fragments of abalone shell that appear to have been used as containers for mixed pigment. An early example of compound tool preparation involving multiple materials and steps. From layers dated to approximately 100,000 years ago, additional evidence of ochre processing and possible symbolic marking has been found. But here is the critical observation that gives rise to the debate about what caused the behavioral revolution. These early instances of symbolic behavior in Africa are isolated, discontinuous. They appear, they disappear, they reappear elsewhere, they disappear again. There is no continuous accumulating tradition of symbolic behavior in the African record from 100,000 years ago to the present. The explosive, consistent, ubiquitous behavioral modernity of the Upper Paleolithic, the tradition that begins around 50,000 years ago and never stops after that, is different in kind from these earlier episodic occurrences. It is as if before 50,000 years ago the capacity for fully modern behavior existed but was not consistently expressed. And after 50,000 years ago, something switches on and never switches off. What was that something? There are several major hypotheses. The first is a genetic mutation hypothesis. The idea that some specific genetic change, somewhere around 50,000 to 70,000 years ago, modified the neural architecture of the human brain in a way that enabled fully modern cognition. Proponents of this view include the archaeologist Richard Klein, who has been arguing for decades that the behavioral revolution was triggered by a specific biological change, possibly related to the evolution of language, that happened in one population in Africa and then spread to the rest of the species. The second hypothesis focuses on language specifically. Language is the ultimate symbolic tool. It allows you to represent absent objects and events, to communicate about things that are not present, to share knowledge across time and space, to coordinate complex social activities, to transmit accumulated cultural knowledge from one generation to the next. A species with language can accumulate cultural innovations in a way that a species without language cannot, because innovations can be described, taught, remembered, and built upon. The threshold for fully modern behavior in this view is the emergence of fully recursive, syntactically complex language, not just communication. Every social animal communicates, but language in the modern human sense. A system with a large vocabulary, complex grammar, the ability to create an unlimited number of new sentences, the ability to embed propositions within propositions, the ability to speak about hypotheticals and futures and pasts and fictions. We cannot directly observe when this kind of language evolved, because language leaves no direct fossils. But the archaeological signatures of fully modern behavior, art, personal ornaments, complex technology, long-distance exchange, are all things that are massively facilitated by complex language. The correlation between the emergence of these behaviors and the emergence of a language-capable neural architecture is at least consistent with the language hypothesis. The third major hypothesis is a demographic one. The archaeologist Adam Powell and colleagues proposed in a paper published in 2009 that the behavioral revolution was driven not by a genetic change or a biological threshold, but by a demographic one. Their argument is that fully modern human cognition had been present since the origin of anatomically modern humans, perhaps since 300,000 years ago. But in small, isolated populations, even individuals with the full cognitive capacity for innovation cannot sustain a complex cultural tradition alone. Cultural complexity is partly a function of population size and connectivity. In a large connected population where ideas flow freely between groups, innovations are preserved, refined, built upon. In a small, isolated population, innovations are lost when the individuals who invented them die before they can transmit them to others. The behavioral revolution on this view was triggered by an increase in population size and population connectivity in Africa, perhaps related to demographic expansion and improved long-distance connections between groups, that crossed a threshold at which cultural accumulation became self-sustaining. There is also a climate connection. The period between approximately 200,000 and 70,000 years ago was climatically challenging in Africa. Multiple cold, dry periods that fragmented populations and reduced population size. The period beginning around 70,000 to 50,000 years ago saw some climate amelioration in Africa that may have allowed populations to expand and reconnect, enabling the demographic threshold for cultural explosion to be crossed. All of these hypotheses have evidence in their favor, and none of them is definitively proven. The truth is almost certainly some combination of all of them, a neural architecture capable of modern cognition, possibly including complex language, possibly evolving in stages over hundreds of thousands of years. Demographic conditions that, when they became favorable, allowed this latent capacity to be fully expressed. And the positive feedback loop of cultural complexity itself, which once started, reinforces and accelerates itself. What we do know, with high confidence, is that by approximately 60 to 70,000 years ago, a population of Homo sapiens in Africa, possibly descended from a single founding group, possibly representing a broader swathe of African populations, began doing something that no hominin had ever done on the same scale before. They started leaving Africa. And this time they did not stop until they had reached every habitable continent on Earth. The out-of-Africa migration, sometimes called out-of-Africa too, to distinguish it from the earlier Homo erectus dispersal, is the most significant human migration event in history. It is the event that produced the modern global population. Every non-African person alive today is descended from a group of Homo sapiens who left Africa approximately 60 to 70,000 years ago. We know this not just from archaeology, but from genetics. Non-African human populations show dramatically less genetic diversity than African populations. This is the signature of a population bottleneck. when a small founding group leaves a larger population and carries with it only a fraction of the original genetic diversity. The founding population that left Africa and seeded all the non-African populations of the world was small. Estimates vary, but some analyses suggest a founding population of as few as a few hundred to a few thousand individuals. From this small founder group, all the billions of non-African people on Earth today are descended. Every European, every Asian, every Native American, every Pacific Islander. But before we can fully appreciate the significance of that migration, we need to acknowledge something else that happened during this journey. Something that no one suspected until the ancient DNA revolution revealed it. When those Homo sapiens left Africa and began spreading across the world, they did not encounter empty lands. They encountered people, different people. And they did not simply replace those people. They merged with them. Chapter 9. The Great Mixing. When our ancestors had sex with other species, the discovery that modern humans carry Neanderthal and Denisovan DNA has required scientists and the rest of us to fundamentally rethink the relationship between Homo sapiens and the other human species it encountered. The traditional model was replacement. Our ancestors arrived, outcompeted the Neanderthals and other archaic humans, and those populations went extinct. Clean, simple, complete. We replaced them. We are us, and they were them, and there is a clear line between the two. The genetics says otherwise. The Neanderthal genome sequencing, completed in 2010 and refined substantially in subsequent years, shows unambiguously that modern humans with ancestry outside of Africa carry between 1 and 4% Neanderthal-derived DNA sequence. The most recent analyses suggest the actual percentage is somewhat variable across different non-African populations, but the Neanderthal signal is robust, consistent, and well characterized. The Denisovan genome sequencing shows that Melanesian and Aboriginal Australian populations carry 4 to 6% Denisovan-derived DNA. Other populations in Asia and the Pacific carry smaller amounts, and there is now also evidence of a third, as yet unidentified, archaic population, sometimes called a ghost lineage or super-archaic, that may have contributed DNA to some modern populations, particularly in West Africa. What this means, biologically, is that Homo sapiens and these other human species were not reproductively isolated from each other. They were interfertile. They could produce viable, fertile offspring. The offspring of a Homo sapiens and a Neanderthal could themselves reproduce, and their descendants' are alive today. This does not mean that Neanderthals and Homo sapiens were the same species. The biological species concept, the idea that two populations belong to the same species if they can interbreed and produce fertile offspring, is actually a simplification, and it breaks down in many real biological situations. Closely related species frequently can and do hybridize when they come into contact, and the resulting hybrids are sometimes fertile. Lions and tigers can hybridize. Wolves and coyotes hybridize. Many pairs of closely related bird species hybridize where their ranges overlap. The fact that Homo sapiens and Neanderthals hybridized and produced fertile offspring does not mean they were the same species. It means they were closely enough related that reproductive barriers had not fully developed between them. They were, in the terminology of evolutionary biology, distinct but incompletely reproductively isolated. Now, when did this interbreeding happen? Where? And what did both sides get out of it? The genetic evidence suggests at least two main episodes of Neanderthal introgression. The formal term for the introduction of genes from one population into another through hybridization, into the ancestors of modern non-Africans. The first and most significant appears to have occurred approximately 50 to 60,000 years ago, probably in the Middle East or Western Asia, as the out-of-Africa migrants first encountered Neanderthal populations. A second episode may have occurred approximately 39,000 years ago in Europe, possibly a short time before the final disappearance of Neanderthals from the fossil record. The Denisovan introgression into the ancestors of Melanesians and Aboriginal Australians probably occurred somewhat later, possibly in Southeast Asia or on the islands of the Pacific, as the ancestors of these populations were making their way to the then-connected landmass of Sahul, the combined Australia-New Guinea landmass that existed when sea levels were lower during the last ice age. What did modern humans inherit from these hybridization events? The short answer is quite a lot of biology that is still functionally important today. Neanderthals had lived in Europe and Western Asia for hundreds of thousands of years before Homo sapiens arrived. During that time, they had evolved local adaptations to the pathogens, climates, and environments of those regions. When our ancestors arrived from Africa, they had no equivalent adaptations to European pathogens or European climates. But by interbreeding with Neanderthals, even briefly, even in small numbers, they could inherit ready-made adaptations, pre-tested over hundreds of thousands of years. Several genes that appear to have been inherited from Neanderthals through introgression are related to immune function. Variants in genes of the innate immune system, particularly genes in the toll-like receptor family, which are among the first responders to bacterial and viral pathogens, show clear signatures of Neanderthal origin in modern non-African populations. These Neanderthal immune variants may have provided immediate protection against European and Asian pathogens that our out-of-Africa ancestors had never encountered before. There are also Neanderthal-derived variants associated with skin and hair characteristics, variants that may have helped our ancestors adapt to lower UV radiation environments outside of Africa. And some Neanderthal-derived variants appear to be associated with sleep and circadian rhythm differences, though the full implications of these are not yet clear. The Denisovan EPAS, one variant that enables high-altitude adaptation in Tibetans, is perhaps the most striking example of beneficial archaic introgression. This specific version of the EPAS1 gene, which regulates the response to low oxygen levels, is found at very high frequency in Tibetan populations and appears to have entered the Tibetan gene pool from Denisovan ancestors. Without this genetic inheritance, the Tibetan people's extraordinary capacity for high-altitude life would not exist in its current form. Not all Neanderthal and Denisovan DNA that entered modern human genomes was beneficial, of course. Some of it was probably neutral, neither helpful nor harmful, simply persisting through genetic drift. And some of it may have been harmful. There are regions of the modern human genome where Neanderthal-derived sequences are notably absent, suggesting that these sequences may have reduced fitness when combined with the modern human genetic background and were removed by natural selection over time. Studies have found possible associations between certain Neanderthal-derived variants and slightly elevated risk for some conditions in modern humans, including potentially some aspects of immune function and possibly some neurological traits. But these associations are complex, and the causal relationships are not fully established. The broader picture that emerges is one of ongoing genetic exchange, not between distinct, cleanly separated species that never mixed, but between related populations that maintained some degree of contact and gene flow, even as they were diverging and adapting to different environments. This picture has forced a reconceptualization of the out-of-Africa model. We did not replace the Neanderthals and Denisovans. We absorbed some of their genetic diversity. We are, in a literal and measurable sense, partly made of the species we displaced. And this leads to perhaps the most profound question in all of human evolutionary biology. If we interbred with Neanderthals, if we could produce fertile offspring together, why are Neanderthals extinct? Why did they disappear? Why are we here and they are not? This is the question the next chapter addresses. Chapter 10. The Last Survivors. Why we are the only human species left. Approximately 40,000 years ago, the Neanderthals disappeared from the fossil record. After inhabiting Europe and Western Asia for approximately 400,000 years, through ice ages, through interglacials, through volcanic catastrophes, the last Neanderthal populations vanished. The timing of their disappearance correlates closely with the arrival of Homo sapiens in Europe. Anatomically modern humans first appear in the European fossil and archaeological record at approximately 45,000 years ago. The last Neanderthals, identified at sites in Spain and possibly Croatia, date to approximately 39,000 to 40,000 years ago. They coexisted with Homo sapiens in Europe for a period of roughly 5,000 to 10,000 years before the Neanderthal archaeological signature disappears entirely. 5,000 to 10,000 years is a long time to coexist. Long enough for the interbreeding events that left Neanderthal DNA in modern humans, but not long enough for Neanderthals to survive. Why not? This is a question that has occupied researchers for decades, and the honest answer is that we do not have a single definitive explanation. We have several plausible contributing factors, and the evidence suggests that all of them may have played some role. The first factor is competition for resources. When two species occupy the same ecological niche, when they eat the same foods, use the same habitats, compete for the same resources, one of them typically outcompetes the other. Homo sapiens and Neanderthals were both medium to large-bodied omnivores occupying the same environments in Ice Age Europe. They were competing for the same game animals. They were using many of the same raw materials for tools. They were sheltering in the same caves and rock shelters. If Homo sapiens had even a slightly higher reproductive rate, even a small advantage in resource acquisition, a slightly higher infant survival rate, a slightly larger social network for sharing information about food locations, the mathematics of population dynamics would, over thousands of years, result in the gradual displacement of Neanderthals without any direct conflict whatsoever. The mathematical model is straightforward. If a Homo sapiens group and a Neanderthal group are both living in the same region, and the Homo sapiens group grows by 1% per year faster than the Neanderthal group, then over a period of 1,000 years, the Homo sapiens population grows by a factor of approximately 20,000, while the Neanderthal population stays roughly constant. The replacement is not violent, it is demographic. The Neanderthal population does not go anywhere. It simply fails to grow while the Homo sapiens population grows around it and into its territory. What might have given Homo sapiens the edge in this demographic competition? Technology is one candidate. The Upper Paleolithic toolkit of Homo sapiens was, by any measure, more varied, more sophisticated, and more rapidly evolving than the Mousterian toolkit of Neanderthals. Upper Paleolithic technology included long blade tools made from carefully prepared cores, bone and antler tools, hafted spears, and possibly atlatls, spear-throwing devices that multiply the force behind a thrown spear, and possibly bows and arrows in the late Upper Paleolithic. Neanderthal technology, while effective for its purposes, did not show the same rapid innovation and diversification. This technological advantage may have translated into more efficient food acquisition, more calories per unit of effort, which would support faster population growth. Neanderthal hunting may have been as effective as Homo sapiens hunting for large mammals, but the diversification of the Upper Paleolithic toolkit, including fishing tools, nets, and small game traps, may have allowed Homo sapiens to exploit a broader range of food resources, providing a buffer when large mammal populations were low. Social networks are another candidate. There is evidence from the distribution of raw materials and shells found at Homo sapiens sites that Upper Paleolithic modern humans were embedded in long-distance exchange networks stretching hundreds of kilometers. Exotic flint from distant quarries, shells from distant coasts, ochre from distant deposits. All of these appear at Homo sapiens sites far from their sources. This implies long-distance social connections, relationships between groups that allowed goods and presumably information to flow across large distances. Information is enormously valuable in a hunter-gatherer context. Knowing where herds of game are currently located, knowing which plant foods are in season in which areas, knowing about unusual food sources, a stranded whale, a localized fruit crop, a seasonal fish run, knowing which techniques and tools work best for which tasks. In a good year, this information network is nice to have. In a bad year, a drought year, a failed migration of the herds, it can be the difference between survival and starvation. If Homo sapiens maintained larger and more integrated social networks than Neanderthals, and the archaeological evidence of long-distance exchange suggests this, then in periods of environmental stress, Homo sapiens populations may have been able to fall back on their wider networks of allies and information sources, while Neanderthal populations, more isolated and more dependent on local resources, were more vulnerable to local food failures. Over many cycles of stress and recovery, this difference in network size and connectivity would translate directly into differential survival rates. Disease is a third factor that has been increasingly discussed. When two populations that have been geographically isolated for hundreds of thousands of years come into contact, they do not just exchange genes, they exchange pathogens. Homo sapiens and Neanderthals had evolved in largely separate geographic ranges, Africa and Europe, Western Asia, for hundreds of thousands of years. Their populations of bacteria and viruses had co-evolved with each population separately. When these populations came into contact, each was potentially exposed to novel pathogens for which it had no immune history. This is the same process, writ large, that drove the catastrophic population collapse of indigenous American populations after contact with Europeans. Diseases like smallpox and measles, to which European populations had some degree of immune history, were essentially novel pathogens for American populations, resulting in mortality rates that in some regions reached 90% or more over the course of a century. Were Neanderthals more susceptible to novel African-origin pathogens that Homo sapiens carried? Were Homo sapiens more susceptible to European pathogens that Neanderthals carried? We do not have direct evidence of disease in the Neanderthal fossil record. Pathogens leave no direct fossil traces. But the possibility that differential disease exposure contributed to Neanderthal extinction is biologically plausible and taken seriously by researchers. A fourth factor is climate. The period from approximately 42,000 to 40,000 years ago, exactly the period during which the last Neanderthal populations disappear from the record, included a series of extreme climate events. Volcanic eruptions in Italy, specifically from the Campi Flegrei volcanic system, produced what is called the Campanian ignimbrite supereruption, approximately 39,000 years ago. This eruption was one of the largest eruptions in Europe in the last 200,000 years. It ejected enormous quantities of volcanic material that spread across Europe and Western Asia, significantly impacting the climate and plant productivity of the region for years to decades afterward. Some researchers have proposed that this volcanic winter, combined with the broader climate instability of the period, delivered the final blow to already declining Neanderthal populations. Neanderthals had survived ice ages before, but they had never done so while also competing with a cognitively and technologically superior invasive species. The combination of climate stress and competitive pressure from Homo sapiens may have been too much. The disappearance of Homo floresiensis on Flores is somewhat less well understood. The youngest Homo floresiensis fossils, approximately 50,000 years old, predate the arrival of Homo sapiens in the region by some estimates, so direct competition may not have been the primary cause. Volcanic eruptions on Flores may have played a role. A change in the local environment, perhaps affecting the stegodon elephant that Floresiensis may have preyed upon, may have contributed. The truth is, we do not have enough evidence to be confident about what ended Homo floresiensis. Homo naledi disappears from the fossil record before Homo sapiens is well established in southern Africa. Its extinction causes are essentially unknown. The Denisovans, as a distinct population, seem to have persisted longer than the Neanderthals. Some evidence suggests Denisovan populations may have survived in parts of Asia until perhaps 40 to 15,000 years ago. Their gradual disappearance as a distinct population may have involved absorption into Homo sapiens populations as much as replacement. The overall pattern, when you look at the last 50,000 years, is one of progressive homogenization. A world that had multiple distinct human lineages, multiple species and subspecies and populations with genuinely different anatomies and possibly different cognitive profiles. This world was being simplified. The diversity was being lost. And what was being left behind was one species, us. But why us? Why did our lineage survive when all the others did not? The most honest answer is probably some combination of slightly superior technology, slightly larger social networks, slightly greater behavioral flexibility, and perhaps some genetic advantages inherited from the archaic populations they encountered. Plus luck. The luck of having been in the right place at the right time. The luck of not having been in the wrong place during a major climate catastrophe. There is nothing inevitable about our survival. There is nothing inevitable about our being the last humans standing. And the sobering truth is this. We are currently living through what is, from a biodiversity perspective, a mass extinction event. The sixth major mass extinction in Earth's history. The only one caused by a single species. Ours. The same characteristics that allowed us to outsurvive every other human species on Earth, our technological capacity, our ability to modify and exploit environments, our demographic expansion, are now driving species to extinction on a global scale at rates that have no parallel outside of the 5 previous mass extinction events, all of which were triggered by geological or astronomical catastrophes. In this sense, the story of the first human is not just history. It is context for the present. It is an explanation of how a species with remarkable cognitive capabilities and remarkable social complexity ended up being not just the only human species on Earth, but the dominant force shaping the entire biosphere. But before we get to the conclusion, before we synthesize everything and answer the question we started with, there is one more dimension of the story that needs to be told. Because everything we have been discussing so far has been about biology, about bones and genes and anatomy and neurological capacity. But the question of what makes us human is not just a biological question. It is a behavioral question. It is a cultural question. And the behavioral and cultural dimensions of humanity took time to fully develop. Time that stretches from the origin of the species at 300,000 years ago to the full flowering of modern behavioral complexity around 45 to 50,000 years ago. Let us now look at what that behavioral revolution actually produced, and what it tells us about the final answer to the question of who the first human really was. Conclusion. What the first human actually was. We have covered 7 million years, from the forests of Africa, fragmenting under the pressure of a changing climate, to the emergence of an upright walking ape on the drying grasslands of the East, from the first stone tools to the conquest of a continent, from a creature with a brain the size of an orange to a species capable of painting horses on cave walls by firelight 40,000 years ago. From a lineage that shared the world with at least a dozen other species of human being to a single surviving species that now numbers 8 billion individuals and has fundamentally altered the chemistry of the atmosphere and the ecology of the entire planet. So let us answer the question, who was the first human? The answer has to acknowledge something that the entire history we have traced makes completely clear. There is no single individual, no single moment, no single species change that constitutes a clean origin point. But there are thresholds. There are moments where the accumulation of changes becomes significant enough that we can point to them as genuine transitions. And depending on which threshold you use to define humanity, you get a different answer. If the threshold for humanity is bipedalism, walking on 2 feet, then the first humans appeared somewhere between 6 and 4 million years ago, in creatures like Sahelanthropus or Ardipithecus that were not human by any other criterion we might care to use. This is too early. Walking upright is a necessary condition for being human, but it is not a sufficient one. If the threshold for humanity is membership in the genus Homo, sharing our generic name, then the first humans appeared approximately 2.8 to 2.4 million years ago, with Homo habilis. Larger-brained than the Australopiths, making stone tools, beginning the dietary shift toward more meat and marrow. This is closer to something meaningful. But Homo habilis was also a creature with a brain less than half the size of ours, probably without language as we know it, certainly without art or complex symbolic culture. If the threshold for humanity is the modern human body plan, the anatomy, the physiology, the skeleton that is recognizably Homo sapiens, then the first humans appeared approximately 300 to 315,000 years ago at Jebel Irhoud and other African sites. Anatomically modern, but with brains not yet fully organized in the modern way, and without the consistent behavioral modernity that we associate with fully human cognition. If the threshold for humanity is behavioral modernity, language, art, symbolic thought, complex technology, long-distance social networks, the full cognitive package that characterizes modern Homo sapiens, then the first fully modern humans appeared somewhere between 100,000 and 50,000 years ago, with the threshold perhaps being crossed definitively around 50 to 45,000 years ago, when the archaeological record permanently changes. And if we use all of these criteria together, if we ask, when did a being exist who walked like us, thought like us, communicated like us, felt like us, made art and cared for the sick and buried the dead and wondered about the world, then the first humans appeared somewhere in Africa, possibly in multiple populations across the continent, sometime between 300,000 and 50,000 years ago, with the most fully modern expression of the species appearing in the record around 45,000 years ago in both Africa and Eurasia. But here is the deepest answer, the one that the whole story has been pointing toward. The first human did not emerge from nothing. It emerged from a lineage of beings who each contributed something. Sahelanthropus contributed the upright posture, or something close to it. Australopithecus developed and perfected bipedalism and demonstrated it could be sustained for millions of years. Homo habilis made the first tools and started down the path of cognitive expansion. Homo erectus built the body, discovered fire, learned to run long distances, and took the lineage out of Africa for the first time. The archaic populations, Neanderthals and Denisovans, contributed immune genes and altitude adaptations that still help their carriers survive today. And somewhere along the line in Africa, a population crossed the final cognitive threshold, developed or fully expressed the capacity for complex language, for symbolic thought, for cumulative culture, and became, in the fullest sense, us. The first human was not a single being. The first human was a process, a long, messy, improbable, contingent process that could have gone differently at 100 points along the way and produced something entirely different, or nothing at all. And what does this mean for us, the ones who survived? It means we carry, in our DNA, in our anatomy, in our behavior, in our social structures, in our instincts and our fears and our loves and our curiosity, the accumulated legacy of 7 million years of experimentation. We carry Neanderthal adaptations in our immune systems. We carry Denisovan altitude genes in our Tibetan relatives. We carry the obstetric consequence of a brain that grew too large for the pelvis in our painful, socially demanding, community-requiring births. We carry the firelit social evenings of Homo erectus camps in our love of gathering around heat and light and telling stories. We carry the Australopith's upright gaze in our instinct to look up, to look out, to look at the horizon and wonder what is there. We are, in the most literal possible sense, the sum of everything that came before us. And the story is not over. In the same way that a Neanderthal living 100,000 years ago could not have imagined the cognitive and cultural world of an upper Paleolithic Homo sapiens, could not have imagined cave paintings and long-distance trade and bone needles and complex language and everything that followed. We cannot imagine what the next phase of this story looks like. What we can do is understand where we came from. And understanding where we came from, truly understanding it, in the deep evolutionary sense, in the geological and genetic and paleontological sense, is one of the most humanizing things there is. Because it reminds us that we are not separate from nature. We are nature. We are the latest chapter in a story that began with the first living cell on this planet billions of years ago. We are the product of an unbroken chain of organisms that for 3.5 billion years never once failed to reproduce before they died. Every single one of your ancestors in an unbroken line stretching back before the first fish, before the first animal, before the first cell with a nucleus, every single one survived long enough to reproduce. Not a single one of them failed. You are the product of 3.5 billion years of unbroken biological success. That is what you are. That is what the first human was, and the second, and all the billions before you, and the billions who will come after. A temporary expression of an ancient ongoing process. A brief, walking, thinking, wondering piece of the universe's matter that has organized itself just enough to look back at its own history and ask, where did I come from? And now you know. Epilogue. The science continues. Everything in this documentary is based on the best available scientific evidence as of the time of writing. And it is important to say explicitly, this field is changing rapidly. Every year, new fossils are found that revise our understanding of the hominin family tree. Every year, new ancient DNA analyses reveal new episodes of population mixing and new genetic contributions to modern human biology. Every year, new dating techniques applied to previously known fossils shift the timeline. Every year, new analyses of known fossil collections using new technologies, micro-CT scanning, geometric morphometrics, proteomics, reveal features that previous researchers missed. The discovery of Homo naledi in 2013 was not anticipated by anyone. The discovery that modern humans carry Denisovan DNA was not anticipated before 2010. The 315,000-year date for Jebel Irhoud was not expected before 2017. The Lomequian tools at 3.3 million years ago were not known before 2015. There will be more discoveries. There will be more surprises. Some of what we think we know today will turn out to be wrong. The picture will get more complicated before it gets simpler. And that is exactly how science is supposed to work. The story of the first human is not a story with a fixed ending. It is an ongoing investigation. New fossils are being found right now in caves in South Africa, and gorges in Ethiopia, and deserts in Chad, and sediments in China, and caves in the Altai Mountains. New genomes are being sequenced from ancient bones found in conditions cold enough to preserve DNA for tens of thousands of years. New analytical tools are being developed that will extract information from fossils we thought were fully analyzed decades ago. The scientists doing this work, the paleoanthropologists digging in remote and hostile places, the ancient DNA specialists working in cold, sterile laboratories, the bioinformaticians analyzing genomes containing billions of base pairs. They are the latest contributors to a tradition of inquiry that stretches back to Darwin and Huxley, and the first recognition that humans are part of nature, part of evolution, part of the biological world. And they are still finding things that astonish them, which means the story of the first human is also, in a very real sense, the story of our own ongoing curiosity about ourselves. Our refusal to accept mythology when evidence is available. Our determination to ask the hardest questions about our own origins and to answer them with rigor and honesty. That determination, that curiosity, is perhaps the most human thing of all. The investigation continues.